Hello, biologists. Mr. B again, and this is going to be our last example from normal two-trait crosses. Coming into the coming days and into the coming weeks, we'll look at sex-linked stuff and some other non-Mendelian genetics. But the test cross is really the last example you need to get through this first packet of practice and the first assignment. So, without further ado, here's what it is asking us. It says, in strawberries, sweet taste is dominant over bitter taste. A strawberry farmer is trying to determine whether his sweet-tasting strawberries are homozygous or heterozygous. This is important, as we'll soon see, because if they're homozygous, the farmer may want to plant with them a lot more than if they're heterozygous, so it's important to know. And this is another one of those problems that requires a bit more reasoning than some of the straight Punnett square ratio questions. So that's why it says explain your answer. It says, how will the farmer be able to do this? Explain your answer. Just to illustrate the difference though, we have two types of strawberries. We have those pretty sweet ones. Then we have these bitter looking sad ones. These ones are sweet and neat, and these ones are not very good. You're welcome. But here's what we know from reading it. We know the farmer has sweet strawberries, and therefore at least one dominant sweetness allele. I'm just gonna say right now, so like we have consistency on the next slide or whatever, we're going to say G is the letter we're using, capital G for good and sweet, lowercase g for not good and bitter, just because S's look the same if they're capital or lowercase. And I trust you can assign alleles by now. But now I want to invite you to pause this video for a second. If we know that the strawberry has at least one dominant allele, what could we cross it with to figure out if the other one is dominant or recessive? That's what a test cross is, and I want to give you a second to think about how you may do that. So yeah, pause the video. 3, 2, 1. All right, welcome back. So these type of test crosses, here, that's what I was saying before. We know the farmer's phenotype has one capital T or one dominant T, so it has at least one sweetness allele. But how can we conclude on the second? And what about if we cross it with a homozygous recessive tester? That could help. If you already came up with that response, good job. If you didn't, don't worry, because it's kind of tricky, and you're not meant to know it already. So, but with that in mind, try both crosses right now before I go to the next slide. Try both scenarios. We're going to do a case 1 and a case 2 deal here, and see if you can figure out what results we'd expect for a homozygous strawberry and what results we'd expect for a heterozygous original strawberry. Take a second and try it out. 3, 2, 1. Oh no, that slide is all broken. Whatever, we'll just let it come up. Okay, so don't look over here just yet. But what we're seeing is we're seeing two scenarios, a case 1 and a case 2 for how our cross could go here. So we're going to cross it with a homozygous recessive tester. Those are these little g's and these little g's. So either way, we're crossing it in the same capacity. But here in scenario 1, if the original strawberry was homozygous dominant, we'll see that it's going to win it's going to mask the recessive in each case. So we're going to have a phenotype of strawberries in the test batch that are all sweet. And that would mean the farmer knew they had a homozygous sweetness berry, and they could go ahead and plant with it. If we crossed it with a homozygous recessive, they would all end up sweet. But if the original berry was heterozygous, see here, then we're going to get a difference where only half of them end up sweet and half of them end up bitter. This tells us we didn't know this allele or this allele before. So by looking at the results, if they all end up sweet, we know that both of them were dominant and they had a homozygous dominant genotype. But if they end up 50-50, we know that the original strawberry was heterozygous because they have to be able to pass on each allele. But remember, for something to show the recessive phenotype, it has to get a recessive allele from either parent. Thus, if there's any recessive phenotype showing up, you know that the parent would have had one present. So here's like the spiel. In case 1, the berry is said to be homozygous dominant. In this case, all the offspring strawberries would be sweet. In the heterozygous case, case 2, half the offspring berries would be bitter, so the farmer is bitter too. More jokes. So basically, this is the explanation. By crossing with a homozygous recessive individual, a test cross allows us to observe a small batch of tester offspring. If all offspring are dominant phenotypes, the original strawberry has been tested to be homozygous dominant. And if only half are each phenotype, or half for each phenotype, then the original berry would have had to have been heterozygous, so it could pass on either type of allele. And that would mean the farmer wouldn't be as happy about planting it. But that's what a test cross is. You're just crossing with a homozygous recessive individual, because essentially that lets um, the unknown individual, the strawberry, uh, do all the talking, because the only possible way to get this bitter strawberry is if there's a bitter allele in the strawberry. So yeah, these are a little bit tricky, but as long as you think about it as it's kind of like a question where you have to come at it from both ways. You come at it with the Punnett square, but then you have to kind of work backwards from the Punnett square and speak to the possible cases. So in this case, we'd know that it was homozygous dominant, but if we got these results, we'd be heterozygous. So until you actually run the test and find out, you can't conclude for sure. But that's what the farmer would be able to do to find out. He could actually run this test and then look at the real children strawberry plants, and if they were all sweet, he's all good to go. And if they were only half sweet, then he had a heterozygous starter. Now, good luck on the assignment tomorrow, biologists. Feel free to come back to these videos at any time and help and use them to help you work through any examples, because the questions you're going to be asked are going to be quite similar. Once again, any questions you have, feel free to email me at mrbsciencemb@gmail.com and send me your assignments when you're done as well. That's all for now, folks. Have a lovely day. Bye-bye.